Hello, my name is Yashir Sagar, and I'll be going through my experiment, which is the effect of varying concentrations of titanium dioxide on the movement of C. elegans roundworms. For the incentives as to why I wanted to conduct this experiment, I wanted to work on a project that relates to biochemistry as something that I want to pursue in college. And after researching for a bit, I found that cell growth and development is something that biochemists do on a daily basis, and it's a big part of their time in the lab. Additionally, I really wanted to work with a live organism that could grow and take care of. For the rationale behind my independent variable, carcinogens such chemicals are widely known as being toxic and being dangerous to human health. One substance that has been in the news lately is titanium dioxide, which is used in food, whitening, and other cosmetics. Titanium dioxide is a white powder that is used in a lot of commonly used human products, and is under scrutiny by food administrations in Europe and in the US through the FDA. It is also argued by some scientists to be completely harmless, while others argue that it is linked to cancer and other hereditary diseases. Titanium dioxide was found to be in Skittles, and I want to see if it has any experimental effects on a live organism. Essentially, I want to see if titanium dioxide is a dangerous component of Skittles. For the purpose of the study was to analyze whether varying concentrations of titanium dioxide affected the movement of C. elegans roundworms. And for the novelty, experiments have been done regarding titanium dioxide and other organisms such as Drosophila melanogaster and Dionario. However, no experiments have been conducted using FDA-approved levels within food. Titanium dioxide have been shown to have effects on the dependent variables in some studies. For example, studies have been conducted on titanium dioxide to learn about its effects and carcinogenicity. Studies have also recorded results documenting oxidative stress, decreased axonal growth in the brain, and decreased locomotor behavior within C. elegans. My hypothesis is that C. elegans introduced to food sources containing titanium dioxide will have less movement compared to C. elegans not introduced to any titanium dioxide via the control group. For the experimental methodology, the independent variables are the varying concentrations of titanium dioxide, which are 0%, 1%, 2%, 3%, and 4%. Additionally, the FDA says that titanium dioxide concentration should not exceed 1% of the food's weight. The FDA guidelines are how I created the values for my concentrations. The model organism for this experiment is C. elegans. And to create a healthy petri dish of C. elegans, agar was made by melting the agar within a nematode growth agar bottle and pouring the liquid agar onto 6 petri dishes. The petri dishes were then left to settle. For the E. coli stock culture preparation, being for the food source, which is E. coli, 0.2 milliliters of E. coli inoculum originating from an E. coli broth culture were pipetted into 2 tubes of E. coli nutrient broth each. The tubes were incubated at 37 degrees Celsius for 24 hours, and then for adding the food source, after 24 hours of incubation, 0.5 milliliters of the E. coli cultures were pipetted into 4 of the petri dishes. To prepare each concentration of titanium dioxide, an empty test tube was weighed and 2 milliliters of the E. coli culture were added to the test tube. The weight of the culture was obtained by subtracting the weight of the test tube with the culture by the weight of the empty test tube, and the percentage of the weight was used to determine the titanium dioxide concentration. The appropriate weight of titanium dioxide was then poured into the test tube. For data collection, 4 quadrants were drawn on each petri dish. Each 1 centimeter cube block housing C. elegans, which were cut from another petri dish, were placed in the same spot for each trial. Data was collected in the opposite quadrant of inoculation, where a 1 centimeter diameter circle lay. A diagram can be found in my paper showing this petri dish setup. Lastly, data was collected on the number of worms that traveled from the site of inoculation to the 1 centimeter diameter circle. Data was also collected on the amount of thrashes recorded within 30 seconds of one randomly selected C. elegans roundworm. All data were collected using a microscope. For the data analysis and results, there were no distinguishable patterns between the petri dishes with varying concentrations of titanium dioxide. The petri dish which contained a 1%, or as shown in the graph, the petri dish which contained a 1% concentration of titanium dioxide had the most worms with 41 compared to 25 worms on the 2% titanium dioxide petri dish, 31 worms on the 3% titanium dioxide petri dish, 38 worms on the 4% petri dish, and 34 worms on the control group or the 0% petri dish. There were no visible outliers, however the error bars were large and demonstrated overlap, signifying inconsistencies within the data. Secondly, the p-value for whether the concentrations of titanium dioxide affected the movement of C. elegans was 0.51, suggesting the use of the null hypothesis, which claims that the data is insignificant. Additionally, there were no significant patterns or trends regarding the torsothoraces of the C. elegans roundworms. As the second graph demonstrates, the petri dish, which contained a 2% concentration of titanium dioxide, had worms who thrashed the most, with an average of 119.25 thrashes, compared to 87.75 thrashes on the 1% petri dish, 116.75 thrashes on the 3% petri dish, 72.25 thrashes on the 4% petri dish, and 109.75 thrashes on the 0% petri dish, the control petri dish. There is no visible trend across the data due to the scattered nature and unpredictability of the data. Lastly, the p-value for whether the concentration of titanium dioxide affected the movement and thrashes of C. elegans was 0.35, again suggesting the use of the null hypothesis, which claims that the data is insignificant. For the interpretations and conclusions, the initial hypothesis of the C. elegans roundworms being affected by the introduction of titanium dioxide concentrations was not supported by the data. This may be down to experimental flaws due to the insolubility of titanium dioxide in the E. coli inoculum, the inability to control the quantity of C. elegans that were added to each petri dish through the 1 centimeter cube blocks, and the difficulty of using the microscope to physically count the worms. The application of this experiment can be expanded and bettered to research how varying concentrations of titanium dioxide affect the movement of C. elegans, using increased standardization of various aspects of this experiment, paired with computational intelligence, computerized imagery within data collection, and more trials. Analyzing the effect that titanium dioxide could have on reproduction may be a viable extension of this experiment. Because of the limitations within this study, a connection between the effect of titanium dioxide on the movement of C. elegans cannot be made. After the data collection within this experiment, there is still no evidence to support the claim that titanium dioxide is not safe for commercial use within consumable products. Thank you for listening to my video.